All right, let's dive right in. Today, we're basically going on a microscopic safari deep inside our own bodies. We're going to meet the trillions of tiny organisms that call us home. But we're not just saying hello. We're becoming microbial detectives, using some pretty incredible science to unmask the secrets hiding in our gut.
And right away, that brings us to this really fascinating puzzle. How is it even possible that the exact same type of bacteria can be a helpful friend in one person, but a seriously dangerous enemy in another? It's like this bizarre Dr. Jekyll and Mr. Hyde situation happening inside us all the time.
So to make this super clear, let's talk about a famous example, E. coli. For most of us, it's a totally normal, even helpful resident of our gut. But, as you can see here, a specific version of it caused this massive, deadly outbreak in Germany. The key difference? It was all in its genetic toolkit. This particular version carried genes for some nasty weapons, like the infamous Shiga toxin. Same species, totally different, and in this case, devastating outcomes. So what's the deal here?
Well, the answer, as we're about to see, lies in looking way deeper than just the species name. See, it's not enough to know what it is. We need to know who it is. So it's time to solve the strain mystery.
So the whole thing hinges on this one crucial concept, a strain. It's basically just a specific genetic version of a microbe. And a great way to think about it, just like we have here, is to compare it to dog breeds. A Chihuahua and a Great Dane are both the same species, Canis familiaris, but you would never ever mistake one for the other, right? They have different traits, different abilities. It's the exact same idea with bacteria.
Which, of course, leads to the next big question. When you're staring at a sample with trillions of bacteria, this massive sea of genetic information, how on earth do you tell these different microbial breeds apart? How do you find that one specific Chihuahua in a park full of Great Danes?
What's so cool about this is how it breaks down the two main detective methods scientists use. First, there's the SNP-based approach. Think of this like proofreading the bacteria's core instruction manual and looking for tiny single-letter typos that make it unique. The other way is to check for gene presence or absence. This is more like looking at the microbes toolkit. Does this strain have the gene for antibiotic resistance while that one over there doesn't? Both of these methods give us a unique fingerprint for every single strain.
Okay, so we've got the tools to identify these unique strains, but knowing how to find them is really just step one. The super exciting part, and what we're going to get into next, is why this all matters. This is where the science literally leaves the lab and walks right into our daily lives.
And this illustrates one of the biggest applications perfectly, your diet. Get this, a study found that the specific strains of a bacteria called Prevotella capri are consistently different depending on whether you're an omnivore or you follow a plant-based diet. Your food choices are literally selecting for different microbial breeds inside your gut, and each one has its own unique skill set.
And for medicine, the implications are absolutely huge. Take fecal microbiome transplants, or FMT. This is where strain-level analysis becomes a clinical game changer. By analyzing the strains in both the donor and the patient, like this diagram shows, scientists can actually predict which donor microbes are most likely to successfully move in and set up shop in the patient's gut. That makes the treatment so much more effective.
This detective work also helps us find microbial superstars. For example, researchers studying children in Bangladesh found this unique strain of Bifidobacterium longum. So what makes it so special? Well, it has the genetic toolkit to digest sugars from both breast milk and the plant fibers you find in solid foods. This is a powerful, newly discovered type of probiotic that could have incredible health benefits for people everywhere.
So up to this point, we've really been focused on the individual players. But the real story of the gut microbiome isn't about individual strains. It's about how they all interact with each other. So now, we're going to zoom out and look at the bigger picture, mapping the entire social network of these microbial communities. And here's the crucial point. Your gut isn't just some random collection of bacteria floating around. It's a bustling, microscopic city. It's a complex ecosystem where all these different microbes are interacting, cooperating, and competing for resources 24/7.
And just like in any city, when the social order breaks down, you get problems. As our source material puts it so well, distortions in these interactions lead to dysbiosis. That's the scientific term for an unhealthy, imbalanced microbial community, which has been linked to tons of different diseases.
So, how in the world do scientists study this complex city? They do it by creating maps, just like the one you're seeing here. Honestly, you can think of it as a social network for your microbes. Each dot is a type of microbe, and the lines show their relationships. This lets researchers see who's friends with whom and how they cluster into different neighborhoods or cooperative groups. Building this map is a really fascinating process. First, scientists measure all the relationships between the microbes. Then, they figure out if those links are positive, meaning they help each other out, or negative, meaning they compete. From there, they can group them into these little cooperative teams, or modules. And by putting all that together, they can finally start to understand how the entire ecosystem works.
Okay, we've gone from the individual microbe all the way to the entire community. So let's bring all of this together and look to the horizon. Because understanding these strains and their social networks isn't just some fascinating scientific puzzle, it is the key that is unlocking the future of personalized health.
The big takeaway here is this. We're moving beyond one-size-fits-all health advice. By decoding the specific strains you have and understanding how they interact, we are entering a whole new age of truly personalized medicine.
And what does this future look like? Well, imagine this. Diets tailored to feed the helpful strains that you personally have. Probiotics that aren't just generic, but are actually designed for a very specific goal inside your body. We could see earlier disease diagnoses just by spotting subtle shifts in your microbial network. And of course, medical treatments that are way more effective and predictable. I mean, that's the ultimate goal.
So that brings us to the end of our deep dive. We've gone from a single species down to a specific strain, and from a single strain to a whole interacting community. It really makes you think. If the unique community of microbes inside of you could tell a story about your health, your diet, and your future, what would you